I'm on. Here we go. So I'm going to say a few things. I'm going to backtrack a little bit about what I said in the previous lecture. And just because I think I'll have time to layer on a little bit more detail. So if you took notes or you have your book in front of you, whatever it is, or nothing, you can just listen. I'm going to backtrack and say a few things about starting with organic compounds. So let me go to my first whiteboard. My iPencil happens to be working today, which is good. The word organic, I think I said this, just means carbon containing. So organic compounds are built with some sort of carbon backbone, usually some sort of carbon chain. And I'm just going to write organic, just always think it contains carbon. Yeah, I got you, Kimberly. Just give me a moment to find you on my list. There you go. So organic, always think contains carbon. Now, inorganic compounds, just so you know, usually do not contain carbon, but a few of them do. There are some exceptions to that rule. For example, carbon dioxide is an inorganic compound that contains carbon, but by and large, inorganic compounds don't have carbon. Organic compounds always do have carbon. When a bond is created between two organic compounds, what we do is, during that process, a water molecule is removed. So I'm just going to write, the removal of water creates a bond. That's called dehydration synthesis, right? And dehydration just means taking away water. Synthesis means to create or make something. So we're creating a bond by removing water. Also, when a bond is broken, that occurs by adding water, especially in organic compounds, except for lipids are the exception. But usually we add water, and that's called hydrolysis or hydrolyzation. So I'm just going to put, what we do is we add water. All right, we hydrolyze it. Let me erase that. Hydrolyze is more proper. There we go. So that's how we put these things together, these organic compounds, and break them apart, is through dehydration synthesis to make something, to create a bond, to make something bigger, a longer chain, or when we need to break up those chains, it's usually by hydrolyzing those bonds by adding water. I'm still admitting students, which is why I'm pausing quite often here. So I did want to briefly explain that, and I know I've said this, I want to hammer it home because I'll expect you to know it for the quiz. That this word monomer, just always think of building block in the context of these four organic compounds. So, for example, the monomer of a carbohydrate is a monosaccharide, a simple sugar. That's the building block for carbs. The monomer for lipids varies. It's a little more complicated. It's usually a glycerol molecule or a fatty acid molecule. The monomer for proteins is an amino acid. The monomer for nucleic acids is called a nucleic acid. And I know I haven't gotten into those details quite yet. All right, so I largely went over carbohydrates last time. I'm just going to add a couple things. So I'm just going to write really quickly, carbs in parentheses continued, because I will have a little bit of extra time to say a little bit more detail about carbohydrates. First, carbs make up about 2 to 3% of body mass. That's not a lot. So our bodies are not made up of very much carbohydrates in terms of relative proportions. I did say this, that carbohydrates include things like sugars, starches, glycogen, and cellulose. Major function, I know I said this, of carbs, I'm just repeating, is energy storage. It's an energy source for the body, mostly to synthesize something called ATP, which I'll get into that detail. So I'm going to rewrite this and I'll tell you why for a reason. So the main function of carbs, I'm just going to write, is a source of energy. And that means to make, our cells make this molecule ATP using carbohydrates as a source. But there's an exception. And the exception to this carbohydrate energy function sort of rule is there are sugars, and they are called deoxyribose and ribose. Those are just two sugars. They're simple sugars. And they are found in our genetic material. So they're found in DNA and RNA. And they have structural functions, not necessarily energy functions. So that's just an exception to but pretty much all other carbohydrates' purpose or function is to use to create energy. I did go over monosaccharides, disaccharides, and polysaccharides last time, so I'm not going to go into those in great detail. Just remember, the monomer of carbs is a monosaccharide. That's a single or simple sugar. And then when we create bonds between those, we remove water to do that, to create a disaccharide or polysaccharides, more than one. And polysaccharides are typically tens to hundreds of sugars linked up together. I think I mentioned glycogen the last time, but the main, if this is a repeat, I apologize, but I just want to make sure that I touched on these. The main polysaccharide in the body, in the human body, is called glycogen. So glycogen is a polysaccharide. That means a big, long sugar. Really what it is stored glucose. So you ingest we all ingest a lot of sugars, carbohydrates, our body breaks them down. We use what we need in the moment. So if let's, for example, you eat a bowl of pasta or you eat a candy bar, the simple sugars that immediately go to your bloodstream are used for energy on demand, whatever it is you're doing at that moment. If we ingest more than we need to use at that moment, then we store the rest of those sugars that we've consumed. And they get stored typically in the liver and the skeletal muscles as this molecule called glycogen. And that's just basically glucose molecules hooked together differently from, say, like a disaccharide. They're hooked together differently. They're stored almost in cartons and containers in our liver. Then we use those stored glycogen molecules as an energy source should we need energy in the future and not have an immediate dietary source. All right. I think I'm done with carbs now. So I'm going to say a few more things about lipids. I believe I covered lipids some on Tuesday. So I'm going to say a few more things about lipids. First, they make up more of the body's mass. So they make up about 18, I'm going to just say 18 to 25%. There's nothing concrete about those numbers. That's just an average. So you can see we're made up a lot more of lipids than we are of carbohydrates. So lipids have what's called a 2-to-1 ratio. That's supposed to be a colon. A 2-to-1 ratio. Sorry, there's a big, I was thinking of carbs. Lipids do not have a 2-to-1 ratio of hydrogen to oxygen like carbohydrates. I'm going to explain the significance of that. Carbohydrates, for every carbon there is in its backbone, in its chain, there's a 2-to-1 ratio of hydrogens and oxygens associated with that. 2 hydrogens and 1 oxygen equals water. So carbs have water molecules basically attached to their carbons. That makes them water soluble. So carbs, I'm just going to write, are water soluble. They dissolve in water. Their bonds can be broken just using water. Lipids are different. You know that fats and water don't mix, that oil and water don't mix. And what that really means is that lipids are not soluble in water. So I'm just going to write, because they have a different ratio of hydrogens to oxygens on their carbons, I'm just going to say lipids are water insoluble. Of course, there's an exception to that rule as well. Some very small lipids, particularly in the blood, they attach themselves to proteins. When they do that, they become water soluble because the proteins are water soluble. But by and large, most all lipids are not water soluble. Okay, and then we started talking about types of lipids. I don't really remember. I didn't rewatch my lecture from Tuesday. I don't know how in detail I got with this, but I'm just going to write types of lipids and mention them. I remember mentioning these to you on Tuesday. Fatty acids are the most simple type of lipid. So there's 2 different types of fatty acids. There's something called saturated, which I'm sure you've heard of, and something called unsaturated. Saturated, chemically, what that means is this. All carbon molecules, so I just wrote a big C, have a single covalent bond with hydrogen. And the key word is single covalent. When a fatty acid is unsaturated, all carbon molecules have double covalent bonds with those hydrogens. That dictates a little bit how they behave, what they can do and can't do. I'm not going to go into that. I just want you to know the difference between what saturated means and unsaturated. I'll circle double covalent bonds as well. Okay, I know I mentioned triglycerides, so I'm just going to layer over this board. Don't worry, I'm recording this. And I'm going to write the second type of lipid. Triglycerides. These, I don't know if I said this or not, but I'm saying it now. These are by far the most common type of lipid in the human body. The building blocks for, I think I did say this, so I'm just going to verbally say this again. The building blocks for triglycerides are glycerol and usually 3 small fatty acids. So just remember the monomers, that means building blocks, of triglycerides are 1 glycerol molecule plus 3 fatty acids. And then when we use the word, when we define something, when we define a triglyceride as a fat, that just means it's in solid form at room temperature. This is pretty intuitive. And something, a triglyceride that we call an oil is just a liquid at room temperature. 3, I did mention phospholipids. They're quite common, not quite as common as triglycerides, but they're fairly common in the human body as well. The reason is because these phospholipids make up every membrane to all of our cells. So I think I said that on Tuesday. I'm just giving you a quick review and adding a few more details on before I get to I'll go through proteins and their structures again as well, but I'm going to layer on a few more details about that. So phospholipids, one thing what they're made of, is a glycerol, not sure I did this or not, plus 2 fatty acids, sorry, F-A-T-T-Y, plus 2 fatty acids plus a phosphate group. And I'm going to go over this in much more detail in chapter 3 after you take your first quiz. But so how we designate, how we draw a picture of a phospholipid is we put that phosphate group that looks like a little balloon head and then 2 tails travel down from that balloon. Those are the fatty acid chains with the glycerol attaching. So I'm drawing several phospholipids lined up next to each other right now. And the phosphate is the round head, and those long chains are the 2 fatty acids, and they're just attached by that glycerol. All right, let's quickly go on to, I'm just trying to repeat, yes, we're number 4, steroids. I said that they're derived from cholesterol. Most of these lipids occur in long chains, like the carbon molecules do this. Yes, they can branch and bend, but they're basically long chains of carbon molecules. Steroids are different. They're derived from cholesterol, and they're ring structures that look like this, that are attached to each other. And it's usually 4 of these. Sometimes there's a tail. So their arrangement, because they're derived from cholesterol, their arrangement it's a ring-like structure, not a chain. I'm going to put rings. All right. I already said that their main contribution, they also contribute to membrane structure. And how we find that is because a lot of times cholesterols are attached to these phosphates on the surface of a plasma membrane. They also contribute, so I'm just going to write membrane. Sorry about the writing, it's a plastic pen on a glass surface. So they contribute to membrane structure, and they make up steroid hormones. We've all heard of steroid. Those are just chemical signals, hormones. The most common ones are testosterone and estrogens. Some people know this, some people don't. Both male and females have both testosterone and estrogens, just in different amounts. So females have higher levels of estrogens, males have higher levels of testosterone. Both are classified as steroids. They're hormones that are classified as steroids. And finally, I know I mentioned eicosanoids. I'm just going to briefly go over that. These are sort of a special class of lipids. They're derived from something called arachidonic acid. So I'm going to write monomer is what they really are, in layman's terms, are signaling chemicals. That's always what I think of when I see the word eicosanoids. I just think, it's a signaling chemical. And the major examples of these are prostaglandins, which I mentioned on Tuesday, and also a class of signaling chemicals called leukotrienes. Prostaglandins are involved in mediating inflammation. They also dilate air passageways. I'm just going to write dilate airways. Prostaglandins is sort of a generic term. It refers to a whole group of signaling chemicals. So when you read the word prostaglandin, it's sort of a generic term. There are specific prostaglandins that do different things. Some mediate inflammation, some dilate airways, others help in pain perception. So for example, when you take something like aspirin to alleviate pain, literally aspirin is a prostaglandin blocker. So prostaglandins help us perceive pain. Leukotrienes, on the other hand, are just largely involved with regulating inflammation in some sort of an allergy sense or infection. I'm not even going to write this out. Leukotrienes are chemicals that are released when we get an infection or an allergen into our system, or sometimes when we get injured. And what they do is they signal certain classes of white blood cells to proliferate and release, like for example, basophils release histamines. Those are dilating chemicals. So leukotrienes are largely involved in signaling white blood cells to regulate the inflammatory process. All right, we're done with that. I'm going to say a few more things about proteins, which is big number 3 in organic molecules. Proteins make up about 12 to 18% of body mass. So they make up more than carbohydrates, but less than lipids. Their building blocks, I already said, are their monomers are amino acids. There's 20 different amino acids in the human body that we know of. You don't have to memorize all 20 of them. That's the good news. I am going to draw really quickly. You're not going to have to draw this on the test, don't worry. But I want you to know that amino acids have 3 sections to them. So this is a generic amino acid. They start with a carbon in the center. That's sometimes called the alpha carbon. So there's a carbon molecule, and there's something called a carboxyl group. I'll write this out, don't worry, attached to that alpha carbon, and it looks something like this. There's an oxygen and an that's called a hydroxyl group, and then there's another double bounded oxygen here. This section of the amino acid that I've drawn right there is actually called the carboxyl group. So an amino acid has a center carbon. I'll label that the alpha carbon, and that means the Greek letter alpha just means the central carbon. Then on this side, there's something called the amino group. So I'm going to draw over to our left a nitrogen. Amines or amino groups always have a nitrogen molecule associated with them, and then 2 hydrogens. You're not going to have to draw this, don't worry. I'll explain to you what's important about this as soon as I'm done drawing it. Let's label this. This is the amino group. And then finally, also attached to this alpha carbon is, it's always designated as an R. And this just means variable chain or variable group, depending on what book you read. It is this that gives each amino acid its specificity. So all amino acids have this same basic structure of an amino group, an alpha carbon, and a carboxyl group. And then it's this variable chain that gives one amino acid its specificity from the other. Right, so that's what an amino acid is. They all have this basic shape and backbone, but then the R variable chain is different in each of the 20 different amino acids. So back to, I'm just going to go to my black pen here. The main functions of proteins. Just going to give you a short list, and I'll give you examples of each of these functions. First and foremost, many proteins are structural in nature. That means it gives our body hardcore structure. Classic example that most books use, there's a protein called collagen. Most of us have heard about that. That is a really strong, tough protein material that's found in things like fibrous connective tissue. We find it in our bones, we find it in our hair and our nails. So many proteins can have a structural function. Some proteins can be regulatory. What does that mean? Specifically, I'm writing hormones. When you take A&P2, you're going to learn that there are two classes of hormones in the human body. Amino acid-based hormones, or protein hormones in this case, and steroid hormones. Those are the two classes. You know that steroids now are lipid-based, cholesterol-based, but there are also hormones that are amino acid-based or protein-based. Proteins can be, functionally, can be contractile. Specifically, there are proteins in our skeletal muscles, in our muscles, that allow our muscles to contract. So that's what that word means. There are also some proteins that can be immunological. Specifically, I'm going to write antibodies. Antibodies are basically really, really small proteins. When we get infected by something like a coronavirus, for example, our immune system recognizes that as foreign, and it creates antibodies. It literally manufactures small proteins to fight that foreign invader. Two more. I'll see if I can fit them over here. Transport is a protein of some functions. I'm going to put in parentheses hemoglobin. Hemoglobin is a protein found in our red blood cells that binds oxygen and transports it in our blood. And finally, the last one, proteins can be catalytic in the form of enzymes. All enzymes are proteins that just speed up metabolism or chemical reactions. I'm not going to go through the 4 levels of protein structure because I don't have anything new to add to that. I will mention, just know what primary level means, what secondary level means, what tertiary level means, and what quaternary level means. I went through that the last time. I do want to say this about, I'm not quite finished with adding a few details to proteins. You might read this in your book. This term, denaturation. basically when I see this word, I think, oh, it's typically it changes the shape or breaks apart a protein, and it renders it, or an enzyme specifically, and it renders it useless. So I'm just going to write this. This is layman's terms, the changing or breakdown of an enzyme, and that causes it to be dysfunctional or non-functional. The most common way to denature an enzyme in the human body is to apply heat to it. So if we raise the temperature in the enzyme's environment, oftentimes that denatures that enzyme and renders it useless. It changes its shape. Sometimes it breaks it apart, but it renders it dysfunctional or non-functional. Another way might be to raise the acidity, make the environment more acidic. That can also denature an enzyme. Last thing I'm going to say about enzymes is how they act. So they speed up chemical reactions. How they do that is by lowering activation energy needed for that metabolic reaction to occur. Classic way to picture this that most textbooks show this is if we're looking at an enzyme reaction in the form of a table, some sort of a table, and we have or a graph, and we put energy on this side of the graph, and this is always a function of time. Say without an enzyme, let's say a particular metabolic reaction in the human body requires this much energy before the reaction proceeds, okay? Needs a lot of energy. When we apply an enzyme to this situation, and I'll explain what that means in a minute. So that's without enzyme. What an enzyme does in picture form, this is still energy needed, and this is still time. The enzyme might do something like this. It causes the activation energy necessary decreases a lot. So the hump to get over in order for a reaction to occur is a lot less. Those aren't my favorite ways of interpreting what an enzyme does. I always picture it more in word form myself, or in a different picture form in which an enzyme binds to a substrate, which is oftentimes another protein, and then it causes usually when an enzyme binds to a substrate, we now call it an enzyme substrate complex. It just means the two of them together. So it causes the enzyme substrate complex to change shape. Sorry about the spelling, I'm trying to write fast. When that enzyme and its substrate changes shape, that's what causes the reaction to speed up. Think about it like this. This is a crude example, but if you have, in order for if you need to unlock a door, here's the doorknob, and here's the keyhole that you stick the key in to unlock this door. If you have a key out here that doesn't fit in here, this reaction just isn't going to occur. What an enzyme does is it changes this shape of the keyed configuration so that this fits. So with an enzyme, the key will fit and we can turn the doorknob and the reaction occurs. That's how I always pictured enzymatic activity. All right, now we are on to number 4, nucleic acids. And that will finish up chapter 2 and I'll be done and I'll take any questions. So I'm just layering over the top of this. The 4th type of organic compound found in the human body are nucleic acids. Specifically, I'm going to write DNA and RNA. I'll define those, don't worry. We've all heard of DNA and now a little bit RNA with this virus thing. So first, the monomer or building block of nucleic acids is something called a nucleotide. And just like amino acids, nucleotides have basic sections that are somewhat similar. Their configuration is similar, but the details are what differentiates between one nucleotide and another. At the end of this, I'll draw a nucleotide and show you the pieces to it. So the monomer are nucleotides. I'll do it now, it'll be easy. Nucleotide consists of these parts. Something called a nitrogenous base, and you can guess it has nitrogen in it. There's also a pentose sugar. Pent means 5, so it's a five-ring sugar. And finally, a phosphate group of some sort. So that's what makes up a nucleotide, are these 3 constituents. Now, DNA stands for deoxyribonucleic that's supposed to be an I, nucleic acid. That's what DNA is an abbreviation for. It's a mouthful, but it's not that hard to understand. Ribo or ribose we know is the sugar, the 5-ring sugar. And the word deoxy just means it's lacking an oxygen. And it's a nucleic acid. RNA is similar, but that stands for, we leave off deoxy, that stands for ribonucleic acid. And so it's ribose sugar, I'm going to write this out, is oxygenated. It has an oxygen incorporated into it. Deoxy, the ribose, I'm going to say lacks the oxygen molecule. We also say that DNA is a double-stranded helix or double helix structure. If I take a sugar molecule, and this is shorthand by the way, I'm not going to draw out a bunch of nucleic acid molecules or nucleotides, but if that's a nucleotide, and I'm just going to color them differently. There's another nucleotide. There's another nucleotide. Here's another one. And I create covalent bonds here. I'm creating a chain of nucleotides. DNA is double-stranded. That means that it's connected to another nucleotide in this fashion. And if you can sort of imagine, we are creating what looks like a ladder with rungs going across the middle. That's what double-stranded means. It means there's two strands or chains hooked up by the rungs of a ladder. Helix means twisting. So if I take this ladder structure and I twist it or coil it, it ends up looking something like this. I'm not going to color code it because it would take me too long to do that. If we twist that ladder into a coil, it kind of comes out looking like this. We've just taken that ladder and twisted it on the ends. That's what DNA actually looks like on high microscopy with high magnification. You know that this is our genetic material found in the nuclei of all of our cells, and we pass, genetic material means we pass on this material from generation to generation and during reproduction to give heritable traits. So on this line, it's a coded message is all it is. And this coded message is what our offspring inherit from both parents in a sort of a mixed up fashion, but we literally get pieces of DNA from our father and our mother incorporated into our own cells, and that gives us our inherited traits. It also, though, is what I call the software for all sorts of things in the human body, particularly protein synthesis. So also on this coded message, not only do we inherit these things, but this code that I'm highlighting with my colored pencil tells a cell what protein to make, how much of it to make, and where to send it in the body. We'll talk about that whole process in chapter 3. But so DNA houses not only the information for our inherited traits, but it also it's the code for things like protein synthesis in the human body. RNA, on the other hand, is simpler than DNA, and it's just a single chain of nucleotides. It's not a double strand, it's a single strand of nucleotides. So this is DNA. What I've drawn down here is RNA. It's simpler, it's smaller, it's not as sophisticated. It does have function in the human body, particularly during the process of making a protein. RNA plays a major role, which I'll describe later. So it's involved in protein synthesis. It doesn't, I don't want to confuse you now. In chapter 3, I'll explain exactly what that means and what that role is. But it also, RNA is also the genetic material in more simple organisms than ourselves, like bacteria and viruses, particularly, like the COVID-19 virus. That virus kind of looks like this, and it has a small piece of RNA in the center of it. That's that viral genetic material. And when we are infected, when our cells get infected by a virus, the virus has a way of penetrating. If this is the wall of a healthy cell in the human body, the virus has a way of penetrating that cell plasma membrane and injecting its viral RNA into our cells. That's how a virus replicates itself. It uses the machinery of our own cells to duplicate itself or replicate itself. The last thing I'm going to say today, and then we're done, I'm going to talk about ATP. Technically, ATP is a nucleic acid. It stands for, the letters ATP stand for adenosine triphosphate. That's where we get the ATP. The molecule itself is considered the energy molecule of all living systems. This is a molecule that our cells spend much of their lives just generating, making, synthesizing. So let's just go over the structure of that molecule and its function. So first, I'll just draw a quick ATP molecule. Don't worry, you're not going to have to draw this yourself on a test. I have a way of doing it that I just memorized when I was young. I draw a 5-ring structure first, and then I add a 6-ring structure to it. So I have a 5 and a 6, and then I attach it to a 5-ring sugar. There's an oxygen, so this is a ribose sugar. So I'll label this stuff, don't worry. These 2 rings that I drew up here are an adenine molecule, okay? And what I've drawn down here is a ribose sugar. Deoxyribose that we find in DNA, it has that ribose sugar, but it does not have this oxygen in the middle of it. That's why it's called deoxy. Nevertheless, this is adenine and ribose. Together, these 2 molecules, these 2 constituents, make up what we call adenosine. So that word literally means an adenine with a ribose. And then finally, triphosphate just means 3 phosphate molecules. So I'm going to attach 3 phosphates, and this is shorthand, out here to the ribose sugar. So that whole thing that I've drawn is an ATP molecule, adenosine triphosphate. So there's its structure. And the last thing I'm going to talk about is its function, what it does. We call it the energy molecule, and here is why. You know what? I'm going to go back to my previous page so I don't have to redraw it. When this molecule is manufactured in our cells, it then gets transported all over the body for use. And what it does is it gives off energy. It's the fuel for our bodies to do things. That all happens right here. When this molecule cleaves or breaks off what's called the terminal phosphate, that just means the last one on the chain. So I'll label that. There's the terminal phosphate. Oftentimes what happens, how this molecule ATP gives energy to other things in the body, it attaches itself to another protein or molecule, whatever it is that needs energy, and then it breaks off, it cleaves this terminal phosphate. The breaking off of that phosphate gives us a picture that looks like this. So now we have this molecule over here is called ADP, which stands for adenosine diphosphate, because it only has 2 phosphates now, not 3, not tri. And then we have this lone phosphate. This is a chemical inertia reaction. So what we're doing is we have a molecule. The original ATP molecule has potential energy, and when it breaks off its terminal phosphate, after it attaches itself to something, it turns that potential energy into kinetic energy. So the breaking of this bond literally transfers energy from this molecule, the original ATP, to the whatever that other molecule is that's attached itself to. Now, just so you know, before I stop talking, if I have an ADP molecule, which is what we have left over because we have 2 phosphates. So after this first phosphate is broken off, there's all sorts of things that can happen. This ADP can regenerate itself. There's different ways to do that in the human body so that it's ATP once again, and that it can keep cleaving off this terminal phosphate. Also, ADP itself can cleave its terminal phosphate here and give off energy, but not as much. So this third phosphate, when it's broken, gives off the most amount of energy. It's the most efficient. If we break off this one, it gives off some energy, but not as much. And likewise, the energy decreases significantly as we break off all these phosphates. So what's most efficient for the body is we start with ATP, it attaches itself to something, it splits, and we get ADP plus P. That gives off a whole lot of energy. And then there are different chemical ways that ADP can be regenerated into ATP. There's something called direct phosphorylation that just sticks a phosphate molecule back on. I'll talk about that when we talk about the muscular system. So I think I'm finished, guys. We're done for the day. If you have any questions for me, let me stop my recording real quick.